An enzyme is a biological catalyst, which means it speeds up a chemical reaction without using itself up, so it can be used again. For all substrates to be broken down, an activation energy level must be overcome. Enzymes overcome this energy barrier by giving an alternative route to increase the substrate's energy, therefore lowering the activation energy needed overall. Most enzymes will work to their optimum rate at 37 degrees Celsius. Enzymes are globular proteins. They have a specific shape due to the sequence of their amino acids. An enzyme molecule is quite large, however a small part of it is functional, which is known as the active site. The molecule which an enzyme acts on is called a substrate. This molecule will fit nicely into the active site to form an enzyme-substrate complex. An increase in temperature means that there is a higher chance that a substrate and an enzyme will collide, so the rate of reaction increases. However, if the temperature exceeds over 40 degrees Celsius, the hydrogen bonds in the enzyme begin to break up, which means the enzymes will lose its structure, so the substrate will no longer fit into the enzyme, and if the temperature continues to increase, the enzyme will not be functionable, so it is stated as a denatured enzyme. The pH of a solution is a measure of its hydrogen ion concentration. Every enzyme will have an optimum pH that they work best at. A change in pH will alter the charges of the amino acids, which make up the enzyme, so it will lose its shape. As a result, the enzyme complex substrates cannot occur. A low substrate concentration means that enzymes only have a little amount of molecules to collide with, which means the enzymes are not working to their full capacity. So the rate of reaction is slow. However, when more substrate molecules are added, the rate of reaction increases as the collision rate for enzymes and substrates has also increased. Once the enzymes reach their full capacity, additional substrates can no longer change the rate of reaction. The lock and key method explains how enzymes work. The lock, active site, has a specific rigid shape, which only allows a specific key, substrate, to fit into it. Scientists have observed that molecules could bind to other sites, so they will alter the activity of the enzyme. So the structure is not rigid, it is more flexible. Enzymes will adjust their shape slightly, so the substrate can fit into them. This makes the enzyme flexible, not rigid. The enzyme has a general shape which is altered in the presence of a substrate. When it alters its shape, it puts a strain on the substrate, which will distort a particular bond, so the activation energy becomes low. Oh!